Bismillahirrahmanirrahim. Assalamu alaikum everyone. Welcome to Stream of Knowledge. Today we will discuss about gram staining. As we have studied in our previous lectures that with the help of gram staining we can divide the bacteria into two categories, gram positive bacteria and gram negative. Although there are some species of bacteria that do not stain with gram stain. For example, Mycobacterium and Nocaria species, as they are acid fast. Now the question is why gram positive bacteria are positive and why gram negative bacteria are negative. In gram positive bacteria, the positive meaning they retain the basic or primary dye called crystal violet, while gram negative do not retain it. Means when after applying crystal violet and iodine complex and then subsequent washing with alcohol, the gram positive will retain their dye and do not decolorize, while the gram positive will decolorize with decolorizer. This is the basic principle. The basic principle is gram positive bacteria stained with crystal violet are not decolorized with acetone alcohol and retain their dye-iodine complex, while gram negative bacteria are decolorized with acetone alcohol and hence take up the counter stain, for example saffron. The difference in staining is due to a difference in the cell wall structure. As we already studied that in gram-positive bacteria there is thick peptidoglycan layer while in gram-negative there is thin layer. As in gram-positive there is thick layer so it retains crystal violet iodine complex while due to the thin peptidoglycan layer in gram-negative the stain wash. Now moving to the procedure. Before studying the procedure, you should know the requirements that are transparent glass slides, wire loop, pipette, cedarwood oil, microscope. The following reagents are also used, and these are crystal violet stain, logule iodine, which act as a mordant. What is mordant? Mordant actually increase the affinity of stain to the cell wall. Iodine combines with crystal violet stain and make a complex, hence the cell wall of the bacteria take it is easily, take it is strongly. The next reagent is acetone alcohol or decolorizer. It decolorizes the dye and the counter stain that may be neutral red or saffron. Now let's study the procedure. The first step is to make the smear. After making the smear, you have to fix it. You can do heat fixation or alcohol fixation. After making smear and fixing, cover the slide smear with crystal violet dye for 30 to 60 seconds. Then wash the stain. After washing, apply Logule's iodine for 30 to 60 seconds and then wash. Then add rapid rapidly decolorize with acetone alcohol and wash immediately with clean water. In the fourth and last step is to apply the counter stain or saffron. Now, what will be the interpretation? If the bacteria in the smear are positive, gram positive, then they take the crystal violet dye and appear purple. And if they are gram negative, although they in this step they also take the dye, but in the subsequent washing, the dye removes. In iodine complex, the crystal violet and iodine complex formed. The major difference which occur between gram positive and gram negative is due to the step 3. In step 3, the gram positive bacteria retain their purple color while gram negative become colorless and they take saffroning or counter stain. After washing the counter stain, let the slide dry and examine the smear microscopically. Now, what are the results? Gram-positive bacteria appear purple or violet under microscope, while gram-negative appear pink or red. Remember, yeast cells, for example Candida, also stain with gram staining. They appear dark purple under microscope. Pus cells, nuclei, and epithelial cells also take up the counterstain and appear pinkish. And that was all about the gram staining. Guys, do subscribe, like, comment, and share. I am a medical student and I have to take my. It really took time to make videos for you. So do subscribe, it will motivate me. Thank you.